Hi, my name is Noelle Frey, and I'm here with Dr. Bijal Shah and Shannon Maude, and we are at the 4th International Working Group for CAR-T cell therapy down here in Tampa Bay, Florida. And we just finished what I thought was a very enlightening and exciting session about CAR-T cell therapy for adults and pediatrics with B-cell ALL. And I don't know about you guys, but I thought the session was so exciting, and I think what's clear is we're all so aware of these initial successes that we've had with CAR-19 products in ALL and how this has really launched this field of CAR-T cell therapy to go after other diseases and other antigens. But what we really saw today, I think, is the next phase of CAR-T cell therapy for ALL. And we saw Dr. Maude talked about using a humanized CAR-T cell product instead of a murine or mouse-based CAR-T cell product, hoping to improve persistence of the CAR-T cells, which would translate into less CD19 positive relapses. I talked, and others did too, about going after more than one antigen on the leukemia cell. And again, the goal there is we have these great remission rates. How can we take it to that next step and improve relapse-free survival? And Bijal, we finally now have an FDA-approved product to treat adults with ALL, and it was exciting to hear that data. And we also heard about an off-the-shelf CAR, which could overcome a lot of the barriers that we face when we use an autologous product. So what's inspired you guys, and what are you most excited about?
Well, I'll start. I mean, I'm excited that I can treat grown-ups. How's that? Yeah. I mean, I never really understood the cutoff of 26 for Tisagenlecleucel. It was always something that I found mystifying, and I understand we have to take the trials and figure out how to bring them forward, but you had published data showing that this could be delivered safely to adults, and I can tell you we did try to deliver it to adults off-label, and we're not successful getting that off the ground. And so it was really, really exciting to finally have a therapy we could bring to adults. I think it emphasized that there's still room to make it better. I think this issue of intention to treat, although I hate the term, it does emphasize that folks can get sick while you're in that phase, trying to get them to CAR-T cell immunotherapy, and at least for me, emphasizes the need to intervene earlier, and ideally with lower tumor burden, where, nicely enough, we still see activity and perhaps even better safety. And so that was really the crux of what I wanted to bring forward, in terms of the allogeneic CAR-T cell products. I have had the pleasure of being able to work with Nathan on this protocol for relapsed refractory lymphomas and ALL, and it's been really cool. We're at a point now where we're seeing expansion that's commensurate with what we would see with an autologous CAR-T cell product, and now we get to do the watchful waiting, right? The hard part. Is that really going to translate into these durable remissions that we want to see in ALL and in non-Hodgkin's lymphoma? And so we're in that process now, but it's been very, very exciting to see that develop and to now have an option that we're actually specifically taking for those post-autologous CAR-T cell relapses.
And I think as well that, now that we've seen these exciting results and now that we have this available across the age spectrum, I think we're all thinking, now what? How can we improve upon this? Because it's been remarkable, but it also brings to light, when we see these remarkable results in a substantial fraction of patients, you want it for everybody. And so it brings to light, the failures of these therapies and how can we improve upon that. And I think that's what I'm excited about too. I think, as you mentioned, getting to the patients who haven't been able to get to this therapy in the past, but then also thinking about the patients who have an initial response, but then have the CAR-T cells fail them by one mechanism or another. I think we're learning a lot about how can we improve upon these therapies and make them last longer, make them overcome and be able to recognize the ways that the leukemia can hide and get around it. And so I'm excited about that, and I think I'm also excited about the prospect of bringing this into earlier lines, as you talked about, and as we have been trying to do in pediatrics as well. And I think we're going to see improvements in those outcomes as we bring this into earlier lines as well.
Yeah, it's very exciting. And Shannon, you have such a large experience, perhaps in the world of these large cohorts of pediatric patients treated initially with what's now Tisagenlecleucel and now with the humanized version of it. And you shared your data today, but what's your instinct about the difference between a murine versus a humanized product? Do you think it's something we should keep exploring, worth that effort?
I do think it is. I think that we have seen that a humanized product can overcome some of the problems that we see in patients who have had poor persistence. But even more importantly, I think if you're starting with a humanized or fully human product, you may run into less of those issues down the road that I think we're starting to see the hints of improved persistence of those products, and perhaps we can see improved safety as well. I think it's a little bit early to know that, and the patient population treated on the trials was different. However, I think, in my view, I think the humanized or fully human products are probably where we need to head to improve upon these therapies. And I think doing them on their own or also in combination is where I hope we're going to be going.
Mm-hmm. That's great.
So, Noel, I wanted to ask you, because I thought your presentation was fascinating, right? That we can come in now with a 19 and a 22 CAR, not only see it prove effective and perhaps improve on that persistence benchmark, but the kinetics of expansion and the fact that you can see one CAR, in a sense, pull up the other. And I'm just fascinated by that. I'm curious, what are your thoughts as it relates to that? And does that also give us some insight into how we should be targeting multiple antigens as opposed to trying to build them all into one cell?
Yeah, so I think it's just a really exciting area in the field right now, and I think next year we're going to have so much more to talk about. And even within this conference, I think we're going to hear a couple other talks in the lymphoma session just about other approaches to this dual targeting. So the approach that we've used in our trials at Penn is to actually manufacture two separate CAR T-cell products, one targeted to 19, one to 22, and infuse them into the patient at the same time. And of course, there's other approaches, manufacturing a product with a single vector with bispecificity or a bicistronic vector, or using two vectors encoding separate CARs in the same product. And I think each of these approaches have strengths and weaknesses. Speaking to our own experience using what I think, as I mentioned, is the simplest method. You make two products and give them to the patient. I think the challenges with that approach, there's logistical and cost challenges. You're manufacturing two products. And what we learned is that you need to think about the two products and their cellular kinetics, how it is when you give them as a single agent, and how is that changed when you give the two products together. For example, if the CAR-19 product we're using tends to have an earlier expansion, how is that cytokine storm that's generated there going to influence that more delayed expansion of the CAR-22, both in terms of efficacy potentially, making that CAR-22 potentially more efficacious, but also in terms of toxicity. And the other challenge that I think we could have predicted, which fortunately we didn't see, is if there's these two products in the body, each kind of going after the same cell, different antigens, is one of those going to be more successful? And as a byproduct of that, the other product kind of dies off, right? And so you lose that potential for dual functionality if one of the CAR T cells just doesn't persist. And I think we'll hear some other talks, but an elegant method of a single vector encoding a CAR with bispecificity. We've learned there's lots of structural issues related to how that CAR is on the surface of the cell that might actually impact the ability of that cell to maintain functionality of both of those antigens. So I think all of the approaches that are under investigation have this potential for great success. And and I think less of comparing different approaches, more in terms of optimizing each approach. And I think in solid tumors, this might be really important too.
And Noelle, it's what I found so fascinating, as you have alluded to, is that we've seen all these different approaches being developed simultaneously. And, in some ways you may think, are we putting in a lot of effort with all these different approaches? But it's really, we've seen that the outcomes that you see are much different, and the behavior of the products are really much different, the way that you do it. And I think what we need to think about going forward, it was mentioned in the Solid Tumor Day yesterday as well, is how can we design clinical trials that we can get a signal from all of these different approaches and sort of pick the winner or figure out what needs to be optimized and developed. I think that's going to be our challenge.
Oh, absolutely.
Well, I certainly look forward to next year. So I want to thank you guys and thank the audience for being with us today.